right, so it's been about 3.5 minutes, 4 minutes. So as you guys are reading, I'm just gonna ask anybody, just whoever wants, just unmute yourself and let's have a conversation about this passage, right? And remember, what we're really trying to do here is not really answer and talk about the cell cycle here, is we're just having this simple conversation here. The MCAT is evaluating whether or not you are prepared to think like a clinician, okay? So whenever anybody's ready, you can either message me in the chat or just unmute yourself when you're ready. And let's just talk a little bit about the 3 things that I asked you to check out about this passage. What you think the main idea is, what you think the objective of this experiment is, and what conclusions you can make based on the table. Come on, guys, anybody.
Okay, I'll do it.
Right.
I'm a little confused, but.
Okay, so what did you think when you read, and just start off by, I know who you are, but introduce yourself to everyone.
Hey, I'm Marshall Franklin. I go to Jackson State. I'm a bio pre-med major.
Okay, all right, cool. So Marshall, let's go ahead and we're gonna read this all together, but real quick, what stood out to you about this passage? What would you say the main topic here is?
I guess the microfilaments. And
microfilaments was one thing you read. What else stood out for you from this passage?
I don't know. I just felt like it was talking about the like the function of the microfilaments.
Really where were they functioning in this passage? What process were they involved in? Did they mention that?
I don't think they mentioned it. But I'm guessing like cell division and stuff.
Ooh, cell division. Let's stop there. Right? So this passage, right, the main idea, main topic about this passage is really the cell cycle. All right. Marshall, how well do you remember the cell cycle? If not, it's fine. You can call a friend and we'll get somebody to hop in and talk to us a little bit about the cell cycle.
A little bit.
A little bit. All right, so let's you and I can go, and whenever you get stuff, we'll see if somebody can help you out, right? So let's start all the way far back, right? Let's start with G0, right? Do you know what G0 is?
G0, is that, no, I don't remember.
All right, so G0 is just when you're resting as a cell, right? Because even though you know that cells replicate, not every cell replicates. Not every cell replicates all the time, right? So your skin cells, right? You've flaked off the skin, right? They're replicating all the time, right? But if you're hanging out, let's say, in a neuron, you're not really replicating that much, right? If you're in a heart muscle, you're never replicating, right? So everyone that's a cell starts off at G0, they're hanging out, and then somebody tells them, okay, start, right? Start replicating, right? So if you're a cell, what comes next? Do you know what phase of cell replication comes after like G0?
Is it G1?
G1, yeah. Do you know what happens in G1?
That's when the cell grows, right?
Grows, exactly, right? So what's the cell's main objective in G1, do you think?
To get all the DNA and stuff so that it can split.
Right, so not so much the DNA, right? Because it's already got the DNA, it's growing, right? It's got to get hefty so that it's big enough so that after it splits in half, right? It's got enough stuff. And more importantly, think about how much energy you got to do. If you had to copy yourself, Marshall, does that sound like something that's easy? It's hard. It's going to take a lot of ATP, right? So a lot of what G1 is about is you're preparing to divide or preparing to replicate your DNA, right? So what comes after G1? S phase. Yeah. So you go S phase, right? So S phase, all you do is you replicate your DNA, right? You just literally copy and paste it, right? Do you know what comes after S phase?
G2.
G2. Do you know what you're trying to do in G2?
So if S phase is replicate G2, is that when you check it, make sure everything is good?
Yeah, you do that actually right after you check, right after, right? But S phase took a lot out of you, so you got to grow some more. All right, so let's just kind of go from the beginning, right? So this isn't even when you start to get through mitosis, guys, right? The reason I took this step backwards is even though this passage is about the cell cycle, it's important for you to understand that the cell cycle, one, doesn't happen all the time. Two, is unique in different cells, right? If you're in your skin versus if you're a brain cell, right? And then three, there's a whole process that occurs before you start the cell cycle, right? First, you have to grow in G1 in order to make sure that you have enough energy and enough supply of everything. S phase, you have to double copy your DNA. And then in G2, you're growing, but here your real emphasis is you need to get enough organelles so that when you split in half, both daughter cells have enough organelles. And then finally, you have to just do a quick check, okay? So thank you so much, Marcel, for helping out with that, okay?
Of course.
All right, awesome. So I wanted to just take that little aside just to do a little bit of content review for everyone that's here. But let's really get back to the meat of our discussion, right? Clinical reasoning and the MCAT. So I'm gonna actually walk you guys through this passage and we're just gonna show you how this is really all about cancer, right? So they first start off by telling you something about the cytoskeleton, and they list in here a number of different functions of microfilaments, microtubules specifically. One of them is mitosis. Specifically, they mention that microfilaments are involved in the transportation of organelles and sister chromatids during mitosis, right? So what they're really saying is that microfilaments are important in the cell cycle, specifically in metaphase and in anaphase, okay? So now that we understand that the first paragraph wasn't really talking about microfilaments, they were presenting us the idea of microfilaments in order to dig in and really, really talk about how microfilaments play a role in the cell cycle. That's something that's super common on the MCAT that can trip you up. I know it might seem that this passage is about microfilaments because they start off telling you so much about microfilaments, right? But as we read along, you'll really start to notice that this passage is really about the cell cycle. So, right? So we're gonna jump ahead and we're gonna say here, we're gonna talk about the experiment. So this experiment was conducted in order to study the function of microtubules and microfilaments in eukaryotic cell cycles, eukaryotic cells. So I'll give you guys a quick hint. Anytime you do an experiment, typically you're gonna either turn something on or turn something off, right? So as I'm reading this, I'm saying, okay, cool, they're planning on turning on and off or stopping the function or turning on the function of microtubules and microfilaments, right? So we're gonna come down here and we're gonna see the results, right? So they are giving a drug, right? And what are they seeing? They're trying to see the observed over time, the changes in morphology and function of the cells, right? So let me bring this all back together. First paragraph, they told us microfilaments play a role in the cell cycle. Second paragraph, they told us they're gonna study how microfilaments play a role in the cell cycle. Third paragraph, they tell us they're gonna run an experiment that has a control, effect A, and two different drugs. And what they're trying to find out is how these two drugs affect microfilaments, but not really microfilaments. They're trying to see how those affect the cell cycle, okay? So I'm gonna just cut to the chase here. This is what that passage is about. This is a picture out of a book called First Aid. It's a step 1 book that you use in med school. And the reason why I pointed out this is a passage that's asking you about cancer is what that passage you were looking at was talking about are these two drugs here, which are microtubule inhibitors. And what they're trying to do is see if you can reason through a small little experiment that's gained at seeing, do you understand that cancer is a cell cycle run amok? And can you think about the fact that any drug you use to stop cancer isn't really gonna kill cancer, it's gonna stop cancer from replicating primarily. And so you might say, hey, maybe if I stop the microfilaments that the cancer drug is using in metaphase and in anaphase, I can stop it from going all the way through to creating daughter cells. That's a clinical reasoning thing that you learn in med school. But as this passage showed you guys that we just did a second ago, the show and tell, this is a passage from Princeton Review. And if you don't believe what I'm saying, look at the actual drug, vinblastine. What does vinblastine do? It arrests cells in metaphase of mitosis. We take a step back and go to the next page, boom. You see the same drug, vinblastine, microtubule inhibitor. This is a whole bunch of drugs about cancer, all right? So I'm gonna just pause here because this first like 15, 20, 30 minutes of this presentation is really meant to be like a weird way of getting you to understand that if you have been studying for the MCAT and you can't break 500, you can't break 507, you can't break 510, you can't get to whatever target score you're looking to get to, and you're working hard every day, you're not a quitter, you know, you're going hard, you did well in undergrad, and you don't understand why you can't get to your score, I can almost guarantee you the reason why is that you don't understand what the exam is about. You're studying content rather than spending time critically reasoning, all right? So now that I've told you guys that, let's actually talk about what you might be doing and why you need to stop it. So first, maybe some of you guys are studying a little too much content, right? That's the most common mistake people do. You're just hitting those Kaplan books hard, hitting those YouTube videos hard, but you're not doing practice questions until you do your full-length exams. You got to do practice questions as much as possible because this is an exam on critical reasoning. And it is a case-based exam, meaning the MCAT, at least the one I took, is not a 59-question exam. It is actually an exam made out of 10 passages that have 6 questions attached to it. The primary learning objective, right, or evaluation competency on the MCAT is critical reasoning, right? If you don't believe me, let's do some math, okay? MCAT passage, anybody? How many questions do we typically see on average on an MCAT passage?
Is it 5 to 7?
5 to 7, right? Let's go 6, right? Somebody else. Anybody heard of a pseudo-discrete? Anybody want to explain what a pseudo-discrete question is? It's not complicated, guys.
It's kind of like you don't need
use information from the passage, but you actually don't.
No, exactly, right? It's a straight up content question, right? On average, there's about 2 pseudo-discretes every passage, right? So Sarah, what percentage of that is that? Like, what fraction would that be?
What percentage did you say again?
So how many questions on an MCAT passage?
6.
How many questions are pseudo-discrete?
How many did you say? 2?
2.
So 30%.
30%, right? Could you be my numbers person, Sarah?
Sure.
All right, so 30% are pseudo-discrete. You're just gonna keep track of everything, okay?
Okay.
Cool. Anybody ever heard the term explicit question? Anybody want to like say what they think an explicit question is?
Something that's just like in the passage and you have to look for.
Exactly. Explicitly stated in the passage, right? Based on the passage, based on the figure, based on the table, according to the passage, what conclusion can be made, right? Do you think you really need to know anything, Fahim, besides like what's on the paper?
No.
No, right?
Not for that.
So let's go ahead and count 2 more, right? So those are 2 explicit questions, right? So Sarah, we have 30%, right? Or 33% was like you needed content, right? Now we're gonna start another bucket. You gotta keep track of all the questions you can answer on a practice passage without any content, okay?
Mm-hmm.
All right, so now we've got 30 in both buckets,
Mm-hmm.
right? Now, what about an implicit question? Anybody familiar with the term implicit questions are?
Isn't that something like we've done right now?
To a certain degree, right? It's like implied, right? And what they're really saying is in cars, it would be called reasoning outside of the text, right? In the science section, it's a little weird, right? What they typically say is they provide you with a fact that is not present in the passage, right? And your objective is to then take that fact and put it in the passage. Figure out how that fact relates to the passage and flow it through the critical reasoning that's presented in the passage and see what pops out, right? So Sarah, how many questions do you think there are that are implicit on each passage? You want to guess?
Is it 2?
2, all right? So do you want to just take a second and do a quick tally, or do you have it? How many questions on an MCAT passage you can answer without and with content?
Without content is a 4 out of, or from the passage specifically is 33% explicit, 33% implicit, and 33% pseudo-discrete.
Awesome. Thank you so much. According to my math, that sounds about right. So I'm going to trust it, right? So thank you so much, Sarah. You can just go ahead and put yourself on mute if you want, all right?
Yep.
So the point I just try to make here is, all right, like let's do the math. We literally said 4 out of 6 questions, 60%, all right, let's go easy numbers. 60% you can answer the questions without even knowing content. 40% you need the content, right? So does anybody not believe that, or anybody want to like challenge that percentage? All right, nobody gonna challenge it? So I'm assuming all of y'all spend 60% of your time doing critical reasoning and practice passages then, if you're taking the MCAT. Is that what I'm hearing?
No.
Why is that?
Just based on things that I've heard about the MCAT, that you need to focus more on content review and, you know, the whole high yield, low yield thing. But I was told by a professor that I had who actually took the MCAT and did really well, that really you just need to be a good test taker more than anything.
Good test taker. That's a good myth. So let's talk about good test taker, right? Good test taker is a myth, right? Good test takers are just people that stumbled upon how the test is written. They just got lucky, right? And today, I just told you step 1 for how you're gonna become a good test taker on the MCAT. It's not about content. It's not a science exam. They don't expect you to be a PhD. They expect you to be able to critically reason, right? So I use this part of the presentation to really stop and get people to reflect, right, on how you're allocating your time, right? How you're allocating your schedule, right? What are you allocating in terms of the activities that you're doing to study, right? The biggest myth I have to tell students, like when I tutor, is look, yes, you do need content, right? But if you got an A or B on the class, you're good to go at the very minimum, right? If you didn't get an A or a B or B in a class, yes, maybe you need to take a little bit of time and engage with the content, really get comfortable with it. But if you got an A or a B, right? What you need to start to do is you need to take content specific, right? Go pick a piece of content that you need to go learn, review it or learn it, go through Kaplan, right? But then don't just sit there. Go do practice passages. Because the MCAT doesn't care that you know the formulas. I tutor MCAT and I don't know half the physics formulas anymore. I can critically reason through what the formulas are about, right? You're not gonna learn that if you pick up the Kaplan book and learn it cover to cover and then go do passages. Because the passages aren't gonna look like the books you read, right? Like, I'm sure some of you guys have had a situation where you get a question wrong on an MCAT question, maybe a full-length exam, and you go back and you Google, you look in Kaplan and Princeton Review and Exam Cracker and you cannot find the answer, right? Or maybe you look at the explanation that AMC gives you and you don't get it, right? That's because it's all about critical reasoning, right? And so we're gonna continue about the things that you guys are doing now and should stop, but like number one thing, right? Like it's all about critical reasoning. So if you're learning physics, for example, right? Does anybody know what the name of the equation that is like, I'll write it out right now because I don't know it. I think it's Q is equal to, I think there might be a delta P in there. Somebody like say the name because I don't know the name of it.
What, flow?
Flow rate, right? What's the name of the, this guy has a, there's a guy's name, like starts with a P or something.
Pascal, maybe?
Pascal, Poisson,
Poussili?
French guy. Let's go French guy, okay?
Yeah, it's like Poussile or something.
All right, cool. This is the perfect, somebody only brought it up because somebody asked me, how do you apply critical reasoning without memorizing to this equation, right? So who's talking? I see an A. Let me get your name and where you're at. The person that just responded was saying something.
Oh, Aziza. And
Aziza.
I'm
Okay, Aziza.
Yeah.
Let's talk through, and I'll teach you how I memorize this equation, and don't even know what it looks like. Okay. So let's start with pressure. Something's got more pressure. Is it going to have a higher flow rate?
If it has more pressure, it has It'll be slower.
I didn't say slower. I said the
Oh,
flow
would
rate.
it'll have a higher flow rate if it has
Flow
that
rate.
pressure. Yeah.
Cool. So in my mind, I say, okay, cool. Delta P has to be at the top, right? Because if delta P goes up, I know Q's got to go up, right?
Yeah.
All right. What else is in this equation? I think viscosity is in this equation, right?
Yeah.
So question for you, honey, water. Which one flows faster?
Water.
Water. So if viscosity goes up, what's going to happen to flow rate?
It goes down.
Down. So it might make sense to put that guy down here, right?
Yeah.
Because if this gets bigger, the other side's got to get smaller, right?
hmm
All right, awesome. What else is in this equation? I literally don't remember. I think there's like a length in there, maybe.
Maybe radius or area. No?
Sure. I don't know. Let's go area. If area was in this equation, right? If the cross-sectional area of a pipe increases, what's going to happen to the flow rate?
It increases.
Increases. So with the if the area is in this equation, is it in the top or the bottom?
The top.
Do you get the point?
Yeah.
I literally do not know this. I got a perfect score on the Chem Phys section. I tutor Chem Phys all the time. I still don't know the equation. It's a physics equation that you actually use in cardiology, which is a unit I'm in. Still don't know the equation because I can critically reason through what actually is going on when the pipe gets bigger. More water can go through, right? What if I put honey in there instead of water? Oh, it's going to flow less, right? That's what the MCAT really wants you to know on physics, right? And it's how they're going to actually give you questions where you can answer without doing the math, right? So I hope that answers your question, Anna. And thank you so much for helping out and answering the questions with me, okay? So let's really go through the last couple things because I want us to actually do passages, right? That's the big thing here. It's not really me talking, it's us working. So don't study too much content, right? But maybe somebody's been sitting here saying, okay, Kamanthi, I do passages, right? But do you review them, right? If you have a 501, guess where the rest of your score points are? They're in the rest of the numbers that you got wrong, right? If you got a 507, what do you got to do to really get better? Go study the mistakes you make, right? Reviewing your exams is super important. Reviewing every practice question is super important, right? But beyond that, you got to actually review them and go in depth, right? Don't do a superficial review, right? What I tell people reviewing a question really means is simple thing. Ask yourself one question. If I got the same question again, how do I make sure I get it right? If I got the same question again, how do I avoid getting it wrong, right? It's not about looking into the past when you review. That's a super small thing, right? You got to look into the future because you got to ask yourself, okay, first, was it a content issue, right? That might be 40% of the questions. Or was it a critical reasoning issue about the other 60%, right? But then when you figure out which one of the two bucket it is, you got to then say, okay, what did I do? How do I avoid doing it in the future? What was I supposed to do? How do I do it in the future, right? That how do I do it in the future is really, really how you get value from the practice passages that you do. Otherwise, you're just kind of making mistakes, looking at your mistakes, nodding at your mistakes, going off and doing more mistakes, right? You never stop and ask yourself, how do I not make that mistake in the future? Next thing, you might like spend time and think about how you're going to avoid these mistakes, but did you ever write it down, right? So like if you don't have like a notebook you keep, a document, an Excel spreadsheet, like if there's no way of you document it, it doesn't exist because you can't make learning insights unless you've written the stuff down, right? Because if you think about it, the only way you make learning insights is you sit back and you look at historical data. You try to find trends, right? I'll tell you a personal trend. I was struggling to kind of get that last little push to hit my target score, right? And I looked and I looked and I looked and I looked. And what I had to benefit me is I kept a really, really nice document of all my mistakes. And so as I reviewed it, I started to realize, oh my God, every single full-length exam I've done, I've missed at least 2 or 3, or sometimes even more, redox questions, right? So that was my learning insight, and it would not have happened if I hadn't kept that stuff documented, right? You get your learning insight, what do you got to do? You got to go and actually figure out what to do next about it, right? So this is really quick. I'm going to go through this part. It's not really much, right? This is how I approach my studying. This is unique to me. Don't like focus as much on this part. Really focus on the part where I've been trying to get you to understand it's a critical reasoning exam and not a science exam, right? So I'm gonna just click through this stuff quickly. So I took a content-specific approach, meaning I would study electrochemistry and then I would go do practice passages shortly after, and then I'd go do flashcards right after, in order to make sure that I consolidated whatever I learned through like my primary knowledge acquisition of the content, right? Flashcards are a great way of making sure that you consolidate the knowledge. Practice passages are a great way to make sure you consolidate the application. Right? Thoroughly document every question that you complete. Review the document or whatever way you keep your stuff written down. Go back on a weekly basis and note any trends that you make, because it's better to fix them in real time and after a week rather than waiting 4, 5, 6 weeks. Excuse me. And then the last thing is kind of related to that. I kind of had a dedicated time where I spent focused on like analyzing my mistakes. I've included a brief outline of how I did this for this type of, for electrostatics that you can use on your own, right? This is like a picture of how I documented the stuff, right? So I've talked to you guys in the last slide about how important it was for me to sort of keep track of what I was doing, because later on, you know, I could go back and say, oh, okay, cool. I need to go work on titration because I keep making those mistakes. Or I might go back and look at my notes and realize, oh, I had a content gap and I needed to go back and review aldehydes and ketones. So little things like these are super, super helpful to kind of help you track those trends, okay? So I think this is, yep, so this is the last part of, that's the last of all of my how to study for MCAT. So I'm gonna pause here, and because what I'm gonna get into next is a lesson, right? So we just finished like the, let me teach or let me talk to you, right? The next part is gonna be let me teach you stuff. So I like to pause here and have a Q&A of about 5 minutes. And I wanna open up to everybody, either in the chat, directly to me, to everyone, or like talking. And let's just talk about the first stuff we talked about, right? So this is where you might ask me like questions about like, is this a resource a good resource over this resource? Should I try this or the other, right? Let's have those MCAT study tip questions right now, because we're getting ready to actually do MCAT, right? So if you don't believe me, that's coming up. We're gonna do that passage and someone's gonna help me answer some of these questions, okay? So 5 minutes, let's just kind of have a dialogue, guys.
Quick question for you.
Mhm.
How do you tell whether something is more so content versus not content?
Right, so it depends on the question, right? So if the question states based on the passage, right, that oftentimes is a explicit question. And, you know, content questions sometimes are very obvious, right? That looks like a content question. But more specifically, I think what you're asking is, how do you know if you missed a question because of content, right? And so what I tell students is when you go back and review, right, number one mistake I notice when people review questions that they got wrong is they go look at the question and then go look at the passage, right? No, you go look at the passage, read it carefully, digest it now that you're not stressed, and then go look at the passage to see maybe there was something that was obvious sitting right in front of me that I couldn't understand, right? Because Fahim, have you done any AMC material stuff?
I've been focusing more on Kaplan and stuff.
Kaplan? Anybody, the reason I'm asking this question, I want to kind of see what people think about the AAMC question explanations. I'll make a point out of it. Anybody like them?
No.
No? They're horrible?
They're very
horrible.
limited.
Go ahead, give me the, let's get in the chat. Let's hear in the chat. I got a horrible, and you can tell me why too, and you can talk. Let me hear it. Very limited, horrible, super vague. The answer is the answer because I said so. I haven't used it yet, right?
I've heard real world is better though at explaining
Right.
things.
So here's my hot take. The AMC questions and the answer choices and the explanations specifically are the best thing that you can do for yourself if you want to score in the top percentile on the MCAT. Because guys, what's the MCAT all about? Fahim, told you at the very beginning, critical reasoning. The reason it's difficult to answer them, the reason why they seem vague or horrible, right? Or the answer is because the answer is, right? It's because you didn't realize it wasn't about content. You are used to answers that you see on exams in college or the ones that you see on UWorld that provide you with a content understanding. right? That's UWorld's problem, content. AMC is about, are you prepared to learn clinical reasoning when you get to med school? And so that answer choice, I literally now as a tutor read answer choices and I can tell you why you got the answer wrong. The answer is because the answer is because the passage said the answer was. You never read it. You didn't get the answer because you never stopped to realize, one, it might have been an explicit question. And so the answer is what the answer was because it was explicitly stated in the passage. Okay? I'm hoping that by even answering that question, I'm starting to drive home a big theme in how you should be changing the way you study.
Are you saying that UWorld is not a good resource then?
No, I'm not. I'm saying I love UWorld. I'm saying that there are two competencies that the MCAT expects you to be prepared for. They expect you to have a base of content knowledge in the sciences that are going to be at the level that you need for day one of med school. Not a joke, literally, day one of med school. The second thing is they have to evaluate if you're going to be ready for, depending on where you go to med school, maybe January or the same first day or your second year, but when they start to teach you clinical reasoning, right? You literally take 2 exams every unit in med school. Medical knowledge, clinical reasoning. Medical knowledge, clinical reasoning. Content, critical reasoning. So what I was really getting at is UWorld, the reason why you guys love those UWorld passages or answer choices, and the reason why I love the UWorld passages and the answer choices is that they provide you with a great explanation of the content, right? But there's another way you can get the answer, right? I'm going to show you guys when we do this passage how you can get the answer, no content, okay? So what other questions do we have? UWorld for content, AMC for practice. Yep, that's what I'd say. UWorld for content, AMC for practice.
Sorry.
Go ahead, anyone.
I put it in the chat, but how do you stop yourself from changing your right answers to wrong answers on full length? Because I've been doing that a lot on the science sections, and I don't know why. I don't understand why I don't trust myself enough,
Because, so I'll give you the advice my MCAT tutor gave me when I took it, right? So he said, you want to, yes, read like you're a graduate student PhD with super close active reading, right? But you want to look at the questions and the answer choices like a 4th grader.
Okay.
Right?
Yeah.
So what I mean by that is people that are that young kind of haven't gotten the idea that there's any point of view, there's any reality, they don't get abstract, right?
Okay.
So
yeah.
if it's there in front of them, it exists. If it's not there, it doesn't exist. And so on the MCAT, if you find yourself doing what you're saying right there, the reason you're second guessing yourself is something that's in your head and not on the paper, right? So what I'd advise you is go ahead and change the answer, fine. But you have to find proof on the page somewhere. Not, oh, this thing on the page and then this thing and then this thing. And then if I go by this other thing and then I round about, I can get to the answer. No, right? If honestly you've taken more than 2 or 3 logical steps, you're down a rabbit hole. That's another factor.
Okay.
So main thing, it's got to be on the paper for you to change your answer. Because think about it. First time you looked at that answer, something on the paper made you feel that that was the right thing.
Right.
So why would you change the answer but not go through the same process that you did the first time? That's what I'm getting at.
Yeah. Okay. Thank you. That makes a lot of sense. Thank
All
you.
right, awesome. Is Khan Academy a good resource for practice passages? Yeah, I love them. And one thing I want to get across, right? So when I talk about learning how to prioritize in your study techniques, right? I try to explain to students that there is no good resource. There really is no bad resource. Every resource has a different purpose for every student, right? So for me personally, I like the Khan Academy passages for Psych Soc. I like their questions for Psych Soc. Why? Because they were hard as crap. I didn't do well on them, but they forced me to really work on a weak area, right? I loved their cars passages. Why? Because I needed to work on that. I didn't like their sciences because I was already good on the sciences according to AMC, which is a much more accurate gauge, and I didn't need that extra content level knowledge, right? Because I came at it with strong enough content, and what I needed was critical reasoning and practicing with the section banks and stuff, right? So big point here I'm gonna make for anyone that's asking is UWorld good? Is AMC good? I don't know, nobody does. Everyone who gives you an answer is bullshitting you. Excuse my French, right? It's, okay, what's the priority? What am I trying to get out of this hour or 2 hours that I'm studying, what resource makes the most sense for that? And then like yourself, like if you know yourself and you know, okay, I like texts that are super dense, maybe an exam cracker or sorry, maybe a Berkeley review might be a little good for you, right? Or maybe you're like, maybe I don't really like stuff that's super duper dense and sciencey, and I want something that's a little bit digestible, right? Maybe a Princeton review and a Kaplan is good for you, for your content. I hope the point gets across.
I have a question.
Yes.
Are you able
do
to
you
hear me
mind,
okay?
can we just start a new tradition? Just start off with my name is and my question and like I am where, just so that I at least have some context of who's talking and where they are.
Sure. So like where I'm from, where I'm studying right now, or
Yeah. Where you are right
okay.
now.
So my name is Malaz, and I'm currently a postbac at the NIH. I'm a graduate from Drake University in Des Moines, Iowa. The question that I was gonna ask is, so I noticed that a lot of my mistakes come from the critical thinking part, where if I don't understand the passage right, I'm not able to answer the question right. And so especially on like lengthy passages. So how do you go about reading long passages, and how do you go about like highlighting stuff on these passages? Like what stuff do you focus on, and what stuff try to get out of the passage to answer the questions?
Nice. So I think you might be the transition caller, right? So you asked the perfect question for us to transition then to the second part of the passage, right? So hang tight and let's see if you don't get the answer by the time we're done, okay?
Thank you.
All right, cool. So let's get started. Let's get learning, all right, guys? So if you came here and your goal wasn't to just listen to me talk, and your goal was to learn, we're about to start learning now. And a couple things I'd encourage you to do is for those that are note takers, I'd encourage you to start to take notes. Not the main things that I say, but I'll tell you verbally what are the main things to take away. In addition, if you filled out a contact us form before you attended today, you'll be getting an email with a recording of today's session, as well as a couple free goodies that we've got from all my tutoring time the last couple of months. So with that, let's start to understand the MCAT science passage, okay?
Question for you, sorry.
Yeah, no problem.
More of a personal thing. I don't know exactly if you got my email, because I don't know, didn't show up on my thing as a...
Yeah, you're not the only one. I had a little bit of email issue going on this morning. Apparently some of the emails didn't send, but if anything, message me your email if you feel like you may have not filled out the contact form or may not have gotten an email, okay?
Thank you.
All right, awesome. All right, so let's get started, right? So there are really 3 types of science passage that you're gonna get, right? There is the informational passage, right? Very common, most common rather, in the Chem Phys section. And what's important for you to understand, right? Informational, inform, right? What that means is they're gonna tell you something that is straightforward, right? They're gonna give you a scientific piece of information that is absolutely straightforward. They may even just describe it to you, right? But what they're gonna do is they're gonna present you the experiment that proves or supports that very, very straightforward piece of information you know, right? You might understand electronegativity, right? But then they might give you a passage that requires you to critically reason through an experiment that is really centrally focused on electronegativity, right? We're not gonna talk about the informational passage too much in depth today because we're focused on a biochem passages. So let's hop on over to the next thing, right? So the experimental passage, right? The most dreaded type of passage, right? Super easy. And after today, I hope you guys are gonna see these passages and kind of enjoy them, right? So it's an experiment, right? And like every experiment, it's gonna have the same components that you learned in high school when you learned the scientific method. It's gonna have a main concept. It's gonna have a hypothesis that's being investigated. It's gonna have an outcome that the researcher is interested in. They might even have a way to measure it, right? And ultimately, it's gonna come down to what the independent variable or variables are and how they affect either the outcome or the dependent variable, right? And more importantly, every experiment has to have an experimental procedure and utilize some lab techniques, right? That's all the little pieces that you have in an experimental passage. So we're gonna skip the informational or the persuasive reasoning, and we're gonna jump into my kind of high yield checklist of thinking through an MCAT Biochem passage, but really when you see MCAT Biochem passages, really think experimental passages, okay? So I'm gonna just walk you through slowly, a couple times, how I use this checklist. And then we're gonna use it ourselves, okay? So I got a couple rules, right? This is a checklist. Don't use my checklist as a chronicle order of things you have to do. It's not gonna make sense. It's a set of things that you know when you go look at a passage, you need to get, right? So let me say that again. It is a set of things that you know that before you even look at the passage, you need to know, right? So if you're talking about like what mental change you need to make in how you look at MCAT passages, right? I encourage you never to go unprepared into any MCAT passage. You should be armed, kind of like a doctor is, with a way of looking at the information the patient provides, right? Organizing it in your head, right? And then proceeding after, right? This is just one way of doing it, and it's modular. You can move it around, change it up, but it's all about showing you the importance of making a list, okay? So step number one, it's a checklist. It's really, really meant to help you stay organized. Step two, you're gonna be prepared when you do an MCAT passage because you're not reading the passage. You're kind of like attacking the passage and like a detective searching out for these 7 things, all right? Step 4, we are gonna talk about its working memory, but I'll save that conversation for a little later, okay? So let's get into it. First thing I do when I look at a passage, I say, what is the main topic, right? And I'll just point out the one thing that most people obviously know, a main topic can be a concept, right? But the most challenging things for a student to pick up on the MCAT is the MCAT makes main topics that are processes, right? It could be a set of steps, right? And so you can't necessarily capture the whole main topic in one word, right? It's not receptor, it's not metabolism. A lot of times with experimental passages, especially the hard ones, right? They're hard because it's a process. It's a set of steps that you have to follow, right? So for example, right? Is it okay with you guys if I make up an MCAT passage?
Yes, please.
Okay, cool. So I'm gonna make up a main topic for an MCAT passage, right? So I think metabolism seems like something that is important, right? And oh, here we go. So I actually ate some yogurt earlier and it was like probiotic. So let's go gut bacteria, all right? And let's see how we can relate them to metabolism. We're gonna make this up, okay? So, you know, maybe you've got bacteria that lives in your gut, right? Lives in your gut. And let's see, let's add some more biochemistry. All right, so gut bacteria, cool. So anybody knows why we can't digest fiber? Just shout it out. Like, why can't we digest cellulose? Go ahead, Allison. I think I saw you unmute yourself for a little bit.
Isn't it a beta linkage?
Ooh, yeah, beta linkage, right? Beta glycosidic linkage, right? So let's make it where these gut bacteria that we have are important for breaking down fiber, because we can't. And let's say after they break it down, they are gonna, let's say, produce, see, metabolism. Let's say they produce short chain fatty acids, okay? I'm like, so I'm gonna go from the top. All right, Allison, do you wanna help me out on this? Because you talked, you wanna join me in on making up this MCAT passage?
Sure.
All right, cool. So we've got started from the top, right? So walk me through what you understand so far about this MCAT passage we're making up.
Okay, so your gut has bacteria to help break down the beta linkages in the fiber so you can produce SCFs.
All right, so SCFs, for those who don't know, are short chain fatty acids, okay? Cool. So let's see, remember we said at the top, I said, let's make this a passage kind of about metabolism, right?
Mm-hmm.
So since we started talking about short chain fatty acids, right? I guess those are going to be absorbed, right?
Yeah.
All right, and then like they get absorbed and then they're going to get stored, right? So what cell are they going to be stored in? Like the short chain fatty acid, fat cells, right?
Yeah.
So how about this? Let's make it where like these short chain fatty acids, right? Let's say they are involved in, let's say they activate a receptor, receptor, pardon my spelling. All right, so let's say these short chain fatty acids, right? So after the bacteria broke down whatever fiber we ate, right? The short chain fatty acids activated a receptor that is, let's say it's only found, just to make this easier, right? For us, it's only on fat cells, okay? And what do you want to happen, Sarah, or Allison, sorry, my apologies. Allison, what do you want to happen after that short chain fatty acid activates that receptor on the fat cell? What do you think the fat cell should do? Should it, sounds like, what do you think?
Break
down fat,
Oh
store
yeah,
it,
it should store it.
store it. You know, why don't we make it break it down? Just because I feel like
Okay.
breaking it down is like positive, right? And I'm like,
Yeah.
Sunday, I'm in a positive mood, right? Because it's like,
Okay.
we're going to have him break it down the fat.
Sure.
Just hang in here
Yeah.
with me, okay?
Yeah.
So it's going to activate fat cells' ability to break down fat. So what do you think is going to happen to like whatever organism that this is in terms of like their waistline if this happens?
It's gonna shrink.
It's going to shrink. So what's going to happen to their body weight?
It's gonna go down.
Body weight's going to decrease. Cool. Awesome, awesome, awesome. All right, cool. So now we've made the main topic, right? So Allison, do you see how now, right, this whole process we just put there, you know, could even be described in one paragraph,
Right.
right? Do you want me to try and like make it into one paragraph to just make it a little easier for us to understand?
Sure.
Cool. Let me see if I can. I'm full of party tricks, guys. Let me see if I can do this party trick. Okay, cool. Let's see. All right. So give me a second. I just have to read. I'm not nearly as good at this as it seems. So, all right. So let's say the passage might say something like, in the human intestinal tract, it's inhibited by a bunch of microorganisms, including some kind of gut bacteria that have been implicated in the regulation of metabolism and the development of obesity and diabetes. The breakdown of fiber in the gut by these bacteria produces short chain fatty acids, for example, butyrate or acetate. And these are super important in the host metabolism. And more importantly, they are implicated in the activation of a unique G-coupled protein receptor. And it has been observed that this activation of this receptor is associated with a change in metabolism. Allison, does that sound like a whole paragraph?
Yeah. Sort of.
Does that sound way more meatier than what we wrote?
Definitely.
Right? But would it have been hard to like look at that and be tempted to say one word?
It would be very tempting.
Right? It's a lot harder to do this whole process, right? Because you know what you got to do next now after we've done this whole thing? We now have to figure out what the passage is specifically talking about. What specifically about the main topic are you going to focus on, right? Or another way of saying that is, what's the objective of the passage, okay? So I'm going to tell you what the objective here is going to be. Why don't we focus on this receptor here, right? Does that sound fair if we focus on the receptor?
Yeah.
All right, cool. I'll tell you, honestly, the reason why I picked receptor is I figured a lot of people on the call know something about a receptor. So we're going to do a word association right now. I'm going to say receptor, and I just want you guys all to just, in the chat or out loud, just say things that kind of remind you when you hear the word receptor. I'll go. Bind. Activate, inhibit. What else?
G protein.
G protein. Ligand. Affinity. KD, inhibitors. What else we got? KM, messenger, cyclic AMP, right? So I'm gonna just stop here and kind of explain why I'm saying that, right? Did you guys remember when I mentioned the word working memory at one point? Anybody?
Yes.
Yes. Does anybody want to talk to me about what they think their working definition of the term working memory is, real quick?
The amount of information that you can store and be able to use when called
Right,
upon.
the amount of information that you can be able to store and use. All right. Anybody else want to give me the number? Like, what's the number of distinct items you can keep in your mind at any given time?
5 to 7?
5 to 7. Let's go 7 plus or minus 2, right? Let's go 7, right? So the reason I'm talking about working memory is I'm trying to explain why some of you guys might get what I term the MCAT fog. Okay, so hang here with me. Have you ever done a passage, right? And about halfway through, you just feel dizzy. Like you're like, I don't know if I'm tipsy. I don't know if it just got cloudy in the room, but I all of a sudden cannot even think. Does that ever happen to anybody? Yeah, Allison's up. Okay, yep, got a couple. Yep. So what do you think you overwhelmed there? You overwhelmed your working memory. You tried to inhale the entire passage, right? And so let me teach you one trick to avoid doing that, right? So you see where that blue circle is on the receptor, right? How many people expect the MCAT not to give us like the specific name of a receptor if this was a real passage? Right? They would give it to us, right? Like they'd give us like this protein. Let's make up a name for it, okay? I'm gonna make up a name for this protein, or this receptor, right? From now on, Allison, you're still helping me with the passage, right?
Yep.
From now on, every time I say receptor, until I tell you not to, are you comfortable saying GCPR43?
Sure.
Okay, cool. And the reason I'm making you do this, all right, and it's gonna be like a teaching exercise, so just like be fun with it, play fun with it, right?
Okay.
Is the MCAT is gonna give you specific names to things, right? They might give you the name of a receptor, GCPR43, right? They might even go further and tell you GCPR43 minus/minus. Allison, what do you think that minus/minus means?
It doesn't have it.
It doesn't have it, right?
Yeah.
So MCAT fog happens when you overwhelm your working memory. When you try to memorize the receptor, Allison.
Oh, GCPR43.
Right. Instead of just thinking of it as it's a receptor, right? Because
Right.
think about the number of things you all have in your head right now about receptors. Activate, inhibit, ligand, affinity, KM, extracellular, intracellular domain, right? You have all this existing information coded in your brain, but you've never seen the term GCPR43, right? So when you see that, you then have to go on a passage, all right, what paragraph did I see it? What context did I see it in? And oh crap, am I running out of time? Am I tired? And all the other things you got to think about, right? So an important it's a trick. It's not really a trick. It's a basic thing that you naturally do in the rest of your life, right? You code out things, not by what they're called, but what they do, what they represent. Is that making sense, Allison?
Yeah.
All right, awesome. So let's get back to our passage, okay? So now that we have this idea of this receptor, and you see that circle part, right? We got through step 2. Right? So guys, remember, for anyone that's following along, it might be easy to get lulled into the conversation I'm having, right? You got to remember, what are we trying to do? We're trying to understand how to attack MCAT experimental passages by basically making one up. And the first thing Allison and I did is we first picked a main topic, and we made sure that that main topic was a process, okay? And then after we made a process, I then picked where to specifically focus on during that passage, okay? So now that we've gotten through that part, let's go ahead and talk about the last little bit, right? Allison, what do you think the most obvious outcome should be on this passage? Or the most obvious way to measure the outcome, either one.
Probably if the person loses weight or not.
So let's go ahead and say the body weight is gonna be our outcome of interest, and we're gonna measure it, let's say in grams. Right? This is body weight, this is receptor, and this is a process, okay? Awesome, so now let's kind of take this all together, okay? So the main idea was about these gut bacteria that break down fiber, which produces short chain fatty acids, which activate a receptor, which is only found on fat cells. And after they do that, those fat cells get activated and they break down all the stored fat, and the outcome of interest is the body weight, which looks like it decreases when this happens, okay? So Allison, now that we've gotten through step 4, you wanna kind of make up some independent variables with me?
Sure.
All right, cool. What's one independent variable you wanna think of?
The breakdown of the fiber.
All right, so let's ask a question, right? So is the independent variable going to be part of the specific part
Oh
of
no.
this? You see that?
Yeah.
Right?
Yeah.
So that's a common thing that happens, right? On the MCAT, MCAT likes to do this thing where like they get you following and reading really carefully, right? And then they just ask you something super basic that you may have not focused on or may have like literally just forgotten, right?
Right.
So for you guys that are listening, don't get caught up listening to me and my voice in my conversation with Allison. It's very enticing. Think through what it is that we are doing. This is a passage that we're breaking down, okay? So Allison, let's go into the circle, the blue circle. Let's pick an independent variable to check in there.
Okay, the receptor. I'm
I'll take
sorry,
that.
the activation of the receptor.
Well, let's even go more basic, right? Because if you think about receptor, right? Maybe receptor. Remember that like receptor, I've said receptor twice.
Oh, GCPR 43.
Remember that time I put the minus on it?
Right.
Maybe, let's say if it's a receptor, that's our independent variable, receptor. Let's make it the presence.
Okay.
Present. You guys get the point. I'm not really great at spelling. And then I'll throw one in there just for, let's just put diet in there because we talked about metabolism and whatnot. You know what? Okay, cool. So let's do a little bit of prediction. What do you predict is going to happen if there is no receptor?
They are going to probably gain weight.
Good. And what do you think would happen if we gave them like a diet that was like super fatty?
If they super high in fat and they don't have the receptor, they're gonna.
Ooh, stop.
Oh.
You just made cardinal rule mistake number one.
Oh no.
Data analysis. It's okay. I'm glad you made that mistake, Allison.
Okay.
You know what? Everybody's about to learn how you look at data, right? So here we go. All right, guys, you listening? So this is a place like I'd actually literally write notes down. So then one thing you can never, ever do is compare two conditions that differ by more than one variable. So what Allison said was was receptor and diet, two variables. All right, so here's how you should always do it. And like I use my hands for this specifically because it's a visual thing that's super easy to get once you see it. So here we go. You can only compare two conditions that differ by one variable because you can look at the two outcomes and say that the outcomes are different or similar, or whatever conclusion you can make can be drawn to the one difference that they had. Let's say it one more time. You can look at two conditions that differ by one variable, right? And that one variable, right, if it's the only one that's there, then you can accurately make a conclusion that whatever the outcome difference is in those two conditions is due to the one variable. All right? You understand, Allison?
Yeah,
All
that
right,
makes a lot of
awesome.
sense.
And you guys that are watching in the back that are not really paying attention, that's literally going to be what we talk about or you do in a practice passage in a second, okay? So we've predicted, we've gone through everything. Do you feel confident, Allison, in just kind of walking everyone through the 6 steps that they should take every time they break down an MCAT Biochem passage?
Sure.
Go ahead.
Okay, so the first thing, well, not in any necessary order, but you need to make sure you know the actual process that is going on and not just a single concept, because it's going to be tempting to say, oh, this is about metabolism, this is about this, but it's often more complicated than that. And you need to know specifically what the main topic is talking about and not just, oh, it's about this. You need to go deeper. And also you need to see what the study is, like what outcome the study is focusing on as well.
Let's take a step back. You got to make sure you find a specific part. Right. Remember that one point where you like jumped up to the breakdown of fiber part?
Yeah.
Right. You want to find the specific part. Okay. So here are some major keys. All right. So if you guys are writing notes, I like to stop here when I have somebody summarize so I can give all y'all major keys. All right. These are words that are going to point you, like literally tell you where this, like the objective of the passage. All right. So these are to determine, to analyze, to hypothesize, to observe, right? To assay, A-S-S-A-Y, which means basically to run an experiment to prove something. I said to hypothesize. Those are some of the big ones, right? What they're gonna basically tell you is, if you think about it, The objective of the passage was to determine it. The objective of the passage was to analyze what? The objective of the passage was around the hypothesis of, right? If you literally memorize those words, you can start attacking passages and not reading them because you'll know what to look for and where to look for it. Okay? So Allison, you want to take us home?
Sure. After you do step number 2, you're going to find out the outcome of interest, like what is a dependent variable, what is being affected, and then you're going to see how it's being measured, and then you're going to find out what the independent variables are, the things that are affecting the dependent variables, and then you're going to predict the outcome and the effects of the independent variables, and then confirm your prediction with the results. Or
okay, cool. So we haven't done the 7th one. We're going to do it in a second, right? But let me ask you guys all a question. Did anybody notice I didn't put outcome or dependent variable before independent variable? And if so, does anybody want to like ask why or know why?
Is it because the dependent variable is the one that's going to be tested on?
Yeah, mainly, but even more like obvious in common sense. Here it is. How many dependent variables do you have in any experiment?
1.
How many independent variables can you have in an experiment?
more than
Many.
one.
Which one would be easier for you to get confused by?
The independent.
Mm-hmm. Where do you typically easily find the dependent variable very obviously?
Near the independent variable. Like
Close though, but think graphically.
oh, on the y-axis.
Mm-hmm. So let's think through that, guys. So there's only ever going to be one dependent variable. You know where to find it all the time. So the reason I point that out is it's a super powerful thing to stop and think about. What's the end point of this whole passage that we did? Body weight. How are you going to measure body weight? In grams. So if I was looking at this passage, I would first go look at the outcome. Remember I told you it's not like you don't have to do it in a row, right? If I told you that the outcome of interest was the body weight of mice, that's going to change how you read everything from the beginning. And it's the easiest thing for you to pick up on every passage because there's only one per experiment and it's right there on the y-axis. And so it's much easier for you to find out. And I like that because if you know where the passage is going to end, it's easier for you to understand it as you read it. Make sense for him?
Yes.
All right. You see how that could be really useful? Like, let's say if you had 5 minutes on a passage and had to triage.
Mm-hmm. Yeah.
All right. I'll show you guys a little bit once we do a couple passages, right? So, Allison, you still here?
Yep.
All right, you ready to do the last thing?
Yep.
All right, here we go. Go ahead and confirm for us on that graph.
Okay.
And go ahead and talk it through, right? So what are you looking to confirm? What did you predict?
So I predict that when they don't have the receptor, their body weight's going to be a lot different than the wild type. It's going to be a lot heavier, probably.
Cool. So let's look at these two. Which one of these two do you want to start with? You want to start with the square or the triangle?
Let's start with the triangle.
The triangle. Okay. And now what are you going to compare that triangle to?
The wild type.
All right.
With the
This
high-fat
one?
diet.
Good. Why is that?
Because if you compare it to the standard diet, it's not going to be correct because they aren't the same
Perfect.
treatment.
Awesome. So the high fat diet. And then we go here and what do we see? You predicted that what was going to be what?
I predicted that when they don't have the receptor, that their body weight's going to be higher, and that is true.
True. Do we need to read anything else or can we continue with the rest of the passage now?
I think we can continue.
Good. What I meant by that is literally, if this was you doing the passage, would you go back and check everything else?
No, because it's been confirmed, but I think.
Bingo, bingo. All right. So, Allison, thank you very much for helping us break down AMC Section Bank Biochem passage number 2. So, congratulations.
It was really helpful for me, so thank you.
All right, awesome. So I hope you guys know that wasn't a fake passage I made up. So that's this passage right now. So I want to pause for a second, and I want you guys to actually go through the real AAMC passage that we just talked through. But I want you guys to do the whole thing, because what we're going to do next is we are going to answer some questions. All right, so I will leave this version up here. I will provide you with my specific breakdown, but I'd like for you guys to just take 5 minutes, or not 5 minutes, let's go till the 6:30 mark, and you guys reread this passage, right? Like, can I get like a like somebody in the chat recognizing that like we just talked about this passage, because I wasn't sure if those connections were made. Can I get a yep, please? Same passage, great, awesome. So I'm gonna go on mute for 5 minutes, give you guys a chance to read it, because I only did the first experiment, and I want you guys to read it thoroughly, because we're gonna do not the pseudo-discrete questions, but we're gonna jump and start with the implicit questions first to really, really see if you understood it, do the explicit questions, and then finish off with the pseudo-discrete. In order to show you that 70% of your exam points and questions do not require content learning, because this is an exam to see if you understand critical reasoning. So 5 minutes and see you at the 30 mark. Hey y'all, I just wanted to see, I didn't get any questions directed to me, so I wanted to see if people were ready for us to move on, or if they prefer to have about 4 more minutes to silently and independently work through the passage.
Can we have more time?
Oh yeah, absolutely. I'll just come back at the 6:30 mark.
Thank you.
All right, everyone. Is it okay if I jump in now? All right, cool, awesome. So here are the breakdowns of the passage. I'm going to leave them here for a second, and I'm going to ask if anyone doesn't mind. Can I get 1, 2, 3, 4, 5 volunteers, please? Just message me, please, your name, just so that I can keep track of who asked. Okay. Got 1, need 4 more. 2. What about the gentleman who asked that great question about how to avoid making mistakes when breaking down passages? All right, let me get 3 more. Got 3. Let me get 2 more. Otherwise, we're just gonna be stuck here. Come on, guys. 3, 4. Okay, 1 last one. There you go, perfect. We got 5 people to volunteer. All right, cool, awesome. So what we're gonna do now is when I call your name or when I message you in the chat, you're just gonna unmute yourself and we are going to talk through the question part. Okay, so I'm gonna go through the passage quickly. I'm not gonna talk about the first half of the question of the passage, except to point out a few things, right? Everybody remember that major key I told you to analyze, right? Another one that I didn't point out is to examine. These are kind of really, really big, easy things that you can pick up on. A few things that I want to point out in the first half of the passage is for all of you that highlight and highlight too much, one great strategy to use with then the MCAT when it comes to highlighting is to make sure that your highlights, whenever possible, begin and end with a verb, because that provides you the ability to capture the action that moves the process forward, right? If you notice, the vast majority of the things that I highlighted either begin with, or for the most part, end with a verb, right? And that's a very, very high yield way of making sure that you highlight and capture the full thought and you capture the action that's moving the process along. Right? So that's the first half, kind of quick tips to pick up on, right? Number 1, when you highlight, start with the verb, end with the verb. Number 2, when you're looking for the objective of the passage, look for spots where you get those key words, such as to analyze or to examine, because they're going to tell you the objective of the passage. So, for example, the first part, the objective was to analyze the contribution of GCPR 43, but remember, guys, we don't say GCPR 43. What do we say, Allison?
Receptor.
Bingo. To energy regulation, right? So again, the word to analyze literally took us to where the passage told us what the objective was. As I mentioned to Fahim, there's only one a dependent variable, the body weight, right? And so if we'd come in and looked at this passage, we would have known that the body weight is the outcome of interest. This passage is about the regulation of metabolism, and everything in between just kind of fills in the picture for us. So if you're looking for a technique to make passages a little bit more digestible, I would do just that. If it has a graph or even a figure like the bottom, you might say, okay, this bottom experiment is about the uptake of glucose, all right? And then you come up here and you realize everything else is just gonna tell you how you got to the uptake of glucose, right? Very, very simple trick, okay? So in terms of breaking down figures, a few things that I wanted to point out to everyone is, the dependent variable is very obvious where you can find it. But sometimes the independent variables aren't as obvious, yet they're standing in plain sight. The independent variables rather, are oftentimes found in a legend. That's a great, great place for you to find the independent variables. And again, if you started off with the checklist and attacked this passage, you could have read it and said, the outcome is the body weight. That body weight depends on whatever this is. If you didn't know, wild type and GCPR was a receptor, right? So it depends on receptor, and it depends on diet, right? Just by a cursory glance at this passage, if we had never even read any of this stuff, right, we could be already able to say like, okay, cool, this passage is about the body weight and whether or not you have like this receptor or this diet. And even if we didn't know what wild type and GCPR are, we could still be able to make some analysis of that relationship there, right? We could say the circle, GCPR 43, seems to be lower than the box, right? In every iteration, without ever having read the passage. And keep in mind, this isn't a technique that stands on its own. It stands because we knew, I know, right? When I read a passage, these are the 7 things I gotta get. I know it's a checklist. So I can start off by reading at the top, or I can come in and look at the figure and say, oh, I know what the outcome is. Let me go read the rest. Proving again, a checklist, not a sequence of events. Okay, so now that we're here, can I see the checklist again? I'm gonna send the slides out, but feel free to write down the checklist. It's not fancy. It's the scientific method, right? It's just been organized a little differently. But remember, it doesn't matter how I organized it. It just matters what you find first and how you navigate the passage on your own. Cool, so now that we've done the first half of this passage, I wanna take a step back and we're gonna talk about the second half, but we're gonna talk about why it's important for you to do content and then go do passages, right? Main reason, you're gonna pick up on very, 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 very obvious patterns, right? So one pattern I'm gonna point out to you that's very obvious is the following. The idea of radio-labeled and the idea of uptake as a way of understanding a cellular process. So I'm gonna give you right now like the straight up experiment that you're gonna see over and over and over on the MCAT. You're gonna have a situation where there's a cell. Inside of the cell, something is happening. Doesn't matter what it is. What is important is whatever's happening inside of the cell depends on the uptake of something from the outside. So if you're a smart researcher or smart MCAT maker, you're gonna radio label whatever it is that you know has to be uptaken into the cell in order to estimate how much whatever cell process is in there is going on. So why does that relate to content and passages? If you learn metabolism and then you go and do metabolism focused passages, you'll notice a very common trend. Radio-labeled glucose in order to understand the metabolic rate inside of a cell, right? It could be a passage focused on the receptor in this experiment, right? But the core method of, okay, the receptor is related to metabolism. An easy way to see how metabolism is running is to label the thing we know has to get in there, glucose, right? Here's another example that's a little bit harder to understand, right? And I'm gonna start off with a question. What is the one base that's unique to RNA?
Uracil.
Uracil. Who said that?
Me, Allison.
Oh, okay, cool. You're gonna be, I guess it's gonna be me and you today. All right, so Allison, if uracil is unique to RNA, right? If I radio label uracil, put it in a bath, and then drop a cell in it, what's the rate of uracil uptake gonna give me an estimate for? Or what can I estimate based on the rate of radio-labeled uracil uptake into this cell?
Transcription?
Bingo, right? If you understood that, you're prepared to pretty much critically reason through almost every single experimental passage that has to do with that concept of an uptake. So when you're reading passages now in the future, Allison, right? And you see uptake, you see radio-labeled, right? Uptake of radio-labeled glucose, right? What the MCAT is trying to see is, can you think through what is radio-labeled? Where does it end up? Meaning, what cellular process is it going to end up in? Radio-labeled uracil can only end up one place. And in that way, you can critically reason through what comes next. Like in that case was transcription. So thanks a lot for helping out, okay? So question time. Fahim, you want to take the first one? You can just take a second, read through it, and then when you're ready and confident, and if you need, you can tell me, go back to the passage. And if you need if the question is one that you're struggling with, you can feel free to like message me in the chat and I'll help you out, or tap you out of the situation if you will. So give it a try, Fahim.
Okay.
We'll work together, right? So I'll read the question, right? So which amino acid is least likely, right? Least likely to be found in the transmembrane domain of whatever this thing is, right? So what do you think they're asking you there, Fahim?
Which amino acids make up the transmembrane, and therefore you can deduce which one isn't.
All right, cool. So which one would be, what properties would you want to not have if you're part of, oh, sorry, which properties do you want to have if you're part of the transmembrane domain?
Hmm.
Do you want to be ionic? Well, let's talk about the transmembrane domain. Here we go. We got these guys. Fahim, what are these guys I'm drawing?
The microtubules.
Not quite. So my pictures are really bad. These are phospholipids.
Oh, okay.
Right. Remember the head and the two tails?
Right.
Yeah. So tell me about the different properties of the heads and the tail. What's the difference between them?
So hydrophilic head and hydrophobic tail.
Hydrophilic head and a hydro what tail?
Phobic.
Hydrophobic tail, right? So this orange part is going to be my transmembrane.
Mm-hmm.
Right. So, Fahim, you notice how you have this close interaction with these tails?
Right.
Right. The tails have what chemical property?
They're hydrophobic.
Hydrophobic. So what do you think the transmembrane is more likely going to want to be?
Hydrophobic.
Hydrophobic, right?
Mm-hmm.
So now that we've understood that hydrophobic tails next to the hydrophobic transmembrane, right? Let's go back to the question. The question asked us, which one is least likely, okay? My trick for doing these kind of questions, I just put true, 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 false on the side and just look and see which one is odd, the odd one out. All right, is it okay if we try to do that?
Sure, yeah.
All right, cool. So before we even do that, what do you think this question really, 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 really is about? Is it even about transmembrane membranes? Is it about that concept, or is it about amino acid chemical properties?
Amino acid chemical properties.
Right, but what was the challenge we had to do first?
Figure out what it is in relation to the transmembrane domain so that we could figure out what it was actually asking.
Right, that's one of the biggest ways that discrete questions are really written. Right? It's all about, they'll give you a situation, because this is a pseudo-discrete question. They'll give you a situation and expect to see if you can break down that one step, oh, transmembrane, it's between the two membrane layers. Okay? So how comfortable are you with your amino acids?
Not so much. I just started biochemistry right now.
Okay,
so
no problem. So you can tap out for this one. Anybody else want to jump in that's really strong on their amino acids? Elad, you want to help me out?
I think that was me. It's Joanna.
Okay, go ahead.
I would say aspartate. I think that's the only one that's charged.
This one is charged. And you said ASP was aspartate, and then what's GLY?
Glycine.
Glycine, and it is what chemical property?
I don't remember.
It's hydrophobic. Because remember, I don't want us to go on. So glycine is hydrophobic. Tyrosine or tryptophan, sorry, and phenylalanine are also hydrophobic, right? The big thing is ASP is charged, so it is least likely to be in there because in the transmembrane domain, you want to be polar. Okay, so high yield tip. One of the best things you can do for yourself for the MCAT, at least the biochem section, memorize all your amino acids, memorize all the 3-letter codes, memorize all the 1-letter codes, and most importantly, understand and be able to apply the chemical properties behind all of the different amino acids. All right, so we've done our pseudo-discrete. I'm sorry, I don't want to try and butcher your name. You were just on. I think you were next.
Joanna.
Is it Yolana?
Joanna.
Joanna.
Yes.
Right, cool. So is there a J or an I?
It's an I.
Okay. Cool. So you want to try this one out?
Sure.
All right, cool. So we're going to do this for this question, right? We're going to read the question, and we're gonna try and categorize it. Is it implicit, explicit, or pseudo-discrete? And then we're gonna try and do it, okay?
Okay.
All right, cool. So for this one, go ahead. I'll read the question first. Which table below shows the expected body weight of wild type and the GCPR43 minus mice after they're fed the high-fat diet and housed for 16 weeks in a conventional condition or a germ-free condition? Germ-free being a condition where the gut does not become colonized. What do you think? Pseudo-discrete, explicit, or implicit?
I think that's an implicit question.
Cool. What do you think is being implied here? What's the implicit part? What part did you not see in the passage?
We didn't talk about the passage didn't talk about a germ-free condition
Okay, germ-free. So what does germ-free mean here?
where the gut is not colonized.
Cool. Do you remember the main topic from when we reviewed the passage at the very beginning? It was Allison and I.
It was talking about the receptor.
Here, let's talk through it. What was the first thing that happened? We had what in our gut? We had bacteria. Oh, I can hear you flipping the pages. Don't worry about the page. Let's talk.
Okay.
All right. So we had bacteria in our gut first, right?
Yep.
We had gut bugs. And then do you remember what those gut bugs did? They broke down fiber. You remember that they broke down the fiber?
Yeah.
All right. And then when they broke down the fiber, do you remember what they created?
The fatty acids.
Perfect. Now we're getting it. So created short chain fatty acids. And then what do those short chain fatty acids do? Do you remember?
They were binding to the receptor.
They activated a receptor. And that receptor was only on fat cells. And when those fat cells were activated, do you remember what happened?
It had to do with the body weight.
And what direction did the body weight go?
It dropped.
It dropped, okay? So you realize how much easier it was for you. I don't know, remember, were you looking at your paper, the second half of this?
No.
But you somehow remembered all of the things that we did. Only because you kept your working memory the first time you looked at this practice passage limited to the most important things. You didn't remember actually all of this as one thing, or one set of a bunch of things. This to you in your mind was just the main idea. And yeah, these first couple points, you needed to get your memory jogged, but you were able to articulate the main topic from one of the hardest practice passages in the AMC section bank, verbatim, live, with 33 people, 15 minutes or so after you saw the passage from memory.
With help.
Right? So what does that tell you about how you should read your practice passages from now on? Are you gonna read everything?
No.
No, we're gonna focus on the main things that can really help us, okay?
Mm-hmm.
All right, cool. So let's get back to the passage, right? So you told me germ-free condition was one the gut didn't get colonized, right?
Mm-hmm.
So how are we gonna connect that? So germ-free. When germ-free is over here, what happens to the right side? What do we not have? In the gut. What do we not have in the gut?
Bacteria.
All right. When we don't have any bacteria, what do we not have?
We can't break down the fiber.
If we can't break down the fiber, what then?
And then we can't make the short chain fatty.
All right. If we can't break down the short chain fatty acids, what can we do that cannot do then?
Can't activate the receptor.
And what's going to happen to the body weight?
It will not decrease.
It will, well, increase, right?
Mm-hmm.
Cool. Now, do you want to try and see if we can find the right graph or the right chart? Now, if you go on your computer, do you see a place where it gives you view options?
Yes.
All right, go to view options, and do you see a spot that says annotate?
Yes.
All right, cool. Go ahead and click that, and you and I are going to annotate through. and figure out how to break down this question. And so for everyone that's watching along, I want you to understand what happened there without you even noticing. She just answered the question. When you're in a germ-free condition, your body weight is going to increase because in a germ-free environment, let's connect the implicit to the passage, you have no gut bacteria leading to no breakdown of fiber, which ultimately leads to no activation of this receptor, right? And if you don't have the receptor, isn't that the same thing as saying, or sorry, if the receptor is not activated, isn't that kind of like saying it just looks like it was never there? What do you think about that? Oh, did I lose you?
No, I'm here.
Do you understand? Do you follow what I'm saying?
Yep.
All right, so let's just keep that in mind, okay? So what are we basically saying here? We're saying if you're in a gut-free environment, you don't have any bugs in your stomach, you're never gonna activate that receptor. So your body weight is basically gonna look as if you never had the receptor.
Right.
So you want us to kind of go through. So let's start with A, right? And I want you to first circle what you're gonna compare first. Walk me through your reasoning as you kind of try to find the answer. Because remember, we're looking for the one that's gonna show us that in the germ-free environment, the body weights went up.
Yeah, so I'm looking at
Go ahead, circle
just
it.
the wild type. And then in the very first one, our body weight for the germ-free environment went up, which is parallel to what we've been saying.
So you're comparing the two, so you're only comparing these two to each other, right?
Yes.
Okay, great. So is that true or not true? Did we expect that?
Yes.
Okay, cool, perfect. And then what are you gonna check next?
Then I would go on, would you check the GPCR in the same graph or no?
This is a great question, right? Great way to ask. So here's two ways of doing this, right? For everyone that's looking at questions like this, and you're trying to think, how do I go through it without confusing myself, right? So one way to do it is say, okay, let me look at all the wild types, and let me just compare with one variable. When I had a germ-free environment, right? I expect the lower number to be bigger. That checks out. That does not check out. That does not check out. And so what I did there is I just only focused on one wild type under one condition.
Yeah, that makes sense.
All right, cool. So now we've eliminated two answer choices. And now we're left with only two choices to consider, okay? So we looked at the wild type condition and we were comparing the germ-free environment. So let's just look at the other side, right? And we expect the bottom number to be bigger or smaller or the same. What do we think?
We expect it to be bigger, the bottom number.
So you're telling me that in the no receptor condition, you expect that if you're in a germ-free, you're gonna have higher body weight than if you're in a situation where you had no receptor and you had a conventional environment, you expect that to be a lower body weight. Is that what you're saying?
Yes, however, I'm just confused with the receptor part.
Perfect. So when the receptor was there, what happened to the body weight?
The body weight dropped when the receptor was present.
When the receptor was not there, what happened?
It went up.
When you were in a germ-free environment, what happened?
It went up.
So would it be fair to say now that the germ-free environment and the no receptor condition are the same thing, ladies and gentlemen? This is the answer, right? And so the final thing, unfortunately, we're at the 7 o'clock mark. The final thing I wanted to kind of show you and explain to you is you have to reason through the question carefully, right? When you had no germs there, right? You told me that was the receptor activated or not activated?
It was not activated.
Okay. And when it wasn't activated, the body weight went up, right?
Mm-hmm.
And when you had GCPR43 minus, or as Austin would say, receptor, this was what you had, no receptor present, right?
Right.
And what happened to the body weight?
It went up.
So if the body weight went up in both conditions, isn't it kind of like saying that, if you don't have the body weight and you don't have a receptor, it's kind of the same because there's no signal after.
Mm-hmm.
Therefore, the answer is this, because you see how it's 57, 55 compared to 52 and 43 here?
Yeah.
The reason being that no signal was sent after. And regardless of whether or not you had a receptor, or regardless of whether or not you had the receptor but were in a germ-free environment or did not have the receptor, you basically look the same, right? And the reason I wanted and always love to end these sessions here is an implicit question is going to ask you to do this, to go from germ-free, right? Remember how we had gut, short-chain fatty acids, GCPR, and then body weight. The hardest questions you're ever going to get, ladies and gentlemen, on the MCAT are going to be implicit questions. Because what they're really trying to see is, can you take germ-free environment and understand it really was another way of them describing to you a condition where there was no receptor there, right? And this is a great place to stop because it's a very difficult critical reasoning question to think through in order for you to just kind of stop and realize that the goal of this lesson was one thing, to show you that the MCAT is not about content as much as you think. It's really about your ability to critically reason through a set of case studies that are trying to see how well you understand and can apply science, okay? So with that, I want to thank you for your time. Thank you so much for dropping in. I'm going to answer a couple of the big questions. Yes, I did record today's session. Yes, I'm going to provide it. I will be sending it out in an email if you filled out a contact form. If not, drop your email right now. In addition, I'm going to go ahead and drop another message in there. So if you enjoyed today's session and want to learn more, that is a link to my website, me and my friend's website. We are all second-year med school students and first-year med school students. I attend SUNY Downstate. A few of our colleagues attend Drexel, University of North Carolina, Chapel Hill, and USC. We are all a collective of pre-med med school students that believe in changing the way medical education and the MCAT are taught. So if you want to learn more, want to have a conversation about your MCAT journey, talk more. If you follow the link, there's a calendar where you can schedule a free 15-minute call with myself or some of the other med school students, probably myself. And it's just very relaxed, get to know you, understand what it is that is on your mind relative to MCAT. And then obviously, if you're interested in actually finding an MCAT tutor, I will click ahead. See if nope, I do not have it. We have our own MCAT tutoring marketplace where med school students, oh geez, guess I hope that's odd. Med school students from around the country with 515s or above, or if they're MS2s who are in a medical education curriculum are eligible to come and tutor. It's all Zoom based and super easy to do. So you got that option if that's something that you've got on your mind. So with that being said, I'm gonna hang until the 7:15 mark and just have conversations with anyone that wants to chat med school or MCAT. And yes, my name is Kamanthi because I am Kenyan. That is a Kenyan colored flag, and these are Kenyan beads for the person that asked.
Thank you so much, by the way.
Yeah, no problem. It was an absolute pleasure.
Are you part of a group that would like answer questions about MCAT prep or like, because I know a lot of people do that too.
Technically a group, I guess. We all answer questions about MCAT, questions about med school when we have time. We tutor MCAT, we mentor. A few of us do more application specific stuff. So we all offer a bunch of different areas of expertise. So if anything, I'm just gonna post again check out. I'm gonna just post the link to our website. There is a diagnostic call in there. So it's a Calendarly. You can just schedule a call with one of us for free to just kind of chat specifically, like where are you on MCAT journey, right? In terms of private tutoring clients, we're only at the moment taking private tutoring clients that have exams that are before September 28th. And we only work with a limited number of students. So I only personally work with about 2 to 3 students who are all taking the exam shortly. So, we try to keep it personal with the people we work with in order to make sure that you guys really get that hands-on touch, because that's what's going to help you get that big jump. And actually, I think one of my students is in here. Alad, are you still in here? He's in here, but he's not chatting. I'm going to text him. But yeah, to answer your question.
And aside from UWorld, do you have recommendations on what to do for content review?
I think for content review, I love UWorld for questions. I think the AMC question packs are great because they are content focused, right? So they're really, really going to make sure that you get the level of MCAT level material, meaning, UWorld is really, really good. I think it's the best one, but outside of UWorld, a lot of stuff, for example, Next Step, great exams. But one of my knock on them is they're super quantitative with exam questions, or with calculations. So I'd recommend YouTube. I love Khan Academy. I love Ninja Nerd. I love the Organic Chemist, or the Organic Chem Tutor, as 3 great resources. I think the books that are provided by Kaplan, Princeton Review, Exam Crackers, Berkeley Review are all quality. Really the challenge is making sure you learn that content and then figure out, AK Lectures is also really good. Learn that content and then apply it. So I just saw Alad, one of my former students, jump in. So I'm gonna let him just chat a little bit about kind of what changing the way he looked at MCAT did for him.
How's it going, everybody? So I actually started working with Kamanthi about a little over 2 months, a month and a half ago.
July 17th to be exact.
Exactly. So I actually took my MCAT end of June, and I was not happy with my result. I did do better than my practices, but still, I was at that 500 mark, not pushing forward. So content was there, but the question was, how was I able to really apply that content to the passages and actually, getting through, right? So we know that overall, as Kamal was mentioning, content is very important, right? But if you don't understand what the passage is telling you, that content in most of the cases can be worthless, right? Because if you don't understand the question, how it applies to the passage, you can know all the content in the world. So what happened was Kamanthi and I met with Kamanthi over Wyzant. We chatted on that website and we started talking. And then he showed me today, he went over the passages and he applied the mapping strategy where you really have to understand the passage before answering the questions. And what happened for me personally is that once I took that extra 5 minutes to really understand all the content in the passage and really all the different variables, independent variables, and how each one affects the other, the questions just flew by. And that's what you want. You don't want yourself looking through between every question for 2-3 minutes and try to find a passage, any nit hole, any pick hole to find, the right sentence. Because overall, those little sentences, they have to all apply to the big picture. So once you get that big picture before going in, everything just flows much faster. And hopefully Kamanthi, as he showed the passages, it just made much more sense. So for me, what happened was once I knew that, I'll tell you, right away I went to the section next to the AMC and just started applying it. Passage after passage after passage, and it worked. Not always, but obviously you got some wrong here and there, and you have to apply some content and learn, and you know it's not always going to be perfect. But the idea is that you have that confidence to understand what the passage is telling you. And with that same confidence, you can also apply that to cars, to chem phys, to biochem, and the psych soc. So what you guys should really practice on is really trying to understand the full picture of a passage, right? To understand the independent variables, what's an inhibitor, what's a stimulator, all of how each one affects the other. And by doing so, you're going to realize that you're going to be answering literally 3 to 4 questions on your on the passage right away. So that's pretty much what I have to say. And I want to also thank Kamanthi at this, with this opportunity for everything he's given me. And I really hope that if you guys apply these little skills, to way you guys can really apply your knowledge, right? Because overall, each one has his own style of learning. But try to figure out what the passage is telling you guys. Because, like at the day in medical school, you're going to get in an article about some enzyme disease, whatnot. You need to understand what they're telling you, right? And that's pretty much an MCAT passage. They're going to be telling you a bunch of random enzymes you've never heard of in your life. But those little words in there, inhibitor, catabolic enzyme, like all these different things, competitive, non-competitive, active site, allosteric site, all these little nitpick points that will overall form the whole picture for you by applying content that you've learned from before. So yeah.
Thanks a lot. Yeah, so really he hit it on the head, right? Like Elad and I had this weird thing where we were looking at one of his full-length exams, and he'd done well, was on pace, and we were looking at the question breakdown and noticed, like he said, he literally, like, the first question might have sat there for like 4 minutes, right? But then after that, it was like 30 seconds, 15 seconds, 12 seconds, 5 seconds, because it's, like I told you, 60%, 66% of the exam is on the paper in front of you. So if you take the time to really read it, like, some of the questions you are going to answer by just making the checklist, right? And then all you got left is the 2 pseudo-discretes, and those are the easiest ones because they're like right there often, right? There was a question that somebody asked about next step. I personally like the exams. The one tiny little knock I have on them is that their chem phys section is a little heavy on calculations, which isn't bad. I'd actually encourage you to go ahead and do them and really, really work through them, grind through them, because MCAT math is it's like low-hanging fruit that if you actually spend the time doing it, could be like very high yield. Also one more big advice, guys, is it's almost like a life thing, right? Don't be afraid of the things that make you uncomfortable. The best way you can see your MCAT score shoot up, even in like 7 days, right? Like I've had students go up like 8, 9 points in 2 weeks, and they're like, oh my God, Kamanthi, you did amazing things. I'm like, no, I didn't do anything good. You just got the confidence to step into those mistakes, right? Into the content areas we don't like. Who over here likes electrostatics? Nobody, right? Who likes electrochemistry? Nobody, right? But who likes fluids? Nobody, right? But I will bet you $100 today that 1 out of those 3 things is going to be on your MCAT passage or MCAT exam whenever you take it, right? I can guarantee you that's going to be a passage. August 7th, first passage on one of the, I think the morning MCAT, fluids, first passage, right? These things are expected, right? And so the best thing you can do for yourself is flip your model of, oh, I'm good at this, I'm getting 85% on this, let me keep doing it to get to 90, and turn into, I'm getting 52% on this, let me get this 52 to 72%, 75%, right? That little change there, right, and taking your 52s to 75s, right, going from the 50th percentile to the 75th percentile on the MCAT produces a 300% chance in your odds of getting into med school, right? And if I can get anything clearer to you, that 300% chance increase in your odds of getting into med school is stuck in all the things you do not want to do. You don't want to do passages because you don't know your content. You don't want to go review physics because it's not your favorite thing. You don't want to do psych soc at the end of the night because you're tired. You forget to do your daily cars passages because it's not fun getting 3s out of 6s on them. You wonder why you can't get from the 50th percentile to the 75th, all right? And so with that, anything else you guys want to know? If not, for those of you guys stuck, go ahead.
I just have a quick question.
Yeah.
I don't know if you have enough time to answer or not. I really like the breakdown you use to read the, like the chemistry and biochemistry passages. It's really helped me. But I was wondering if you can apply that kind of on the cars, because no matter how much I practice on the cars,
Yeah, so
I keep not knowing how to read the cars.
yeah, so I'm not even gonna lie to you. Cars, I'm not a cars specialist. In our groups at EliteMD, I tutor Chem Phys and Biochem and MCAT critical reasoning and coaching. But I have other colleagues, and if you send me an email, I can try and connect you with one of them that one of them got a perfect score on the car section and was an English lit major. So not my wheelhouse, unfortunately.
Would you mind dropping your email again?
Yeah, so if you, instead of my email, so what we do is, because we prefer to cut out emails when we can just schedule the meeting, right? So I'm gonna post again. That is a link to our website. If you go on our website right now, anytime you click here, I'll take you over. If you come here, click the Let's Connect button, it'll take you to, oh wait, wrong one. Book a free tutoring session. It's going to provide you a Calendly link to book. And I think some people have already been booking while we've been talking. So if you want to meet, like for example, if your exam is this month and you're looking to grab time with me, these are the times I'm available, or someone is available, because some of these times are other tutors. Like a cars one, I think, is the 5:30 guy. So just go on the website that I posted, schedule a meeting. It's going to be free.
When did you say the cars one is available?
It's the 5:30 time, I believe, or not the 5:30, 7:30 time, because he's West Coast.
All right, sounds good. I will go on there right now and do it.
Awesome. And do you have an exam coming up?
I actually I don't know yet. I'm planning on doing it sometime in January or February, but I have been studying cars for the past 2 months and I still don't see improvement. So I'm really trying to know like how to read better.
It's a super difficult subject, and I'll tell you this, right? Keep your head up, right? Keep grinding, because it's one of those subjects where if you don't quit, eventually it's going to work out.
Awesome, good to know. Right. Well, again, thank you very much.
No problem. Anyone on the call have exams like September 2020 exams that wants to talk? Okay, I got two.
I'm on September 28th. Okay. I'm scheduling a call, I guess, with you guys now. Only because,
go ahead, book it.
Yeah, I don't know. I just feel like, I don't know if anyone else experiences this, but this like testing anxiety of never like, fully feeling ready. So I'm like, you get over that.
I mean, I'd be lying to you if I told you do get over it. It's all about managing it, right? And like for me, for example, right, I had horrible test anxiety, not in the rest of my life, right? I never had test anxiety, but literally opening, like the blue portal that the MCAT like exam is in?
Yeah.
That used to give me anxiety. And I realized the best way for me to get over it wasn't to kind of like force myself to go sit there and then do a bunch of passages and not do well on it. Like I picked a personal strategy, which was, I bought a hard copy book and did practice passages there. And by the time I was like, okay, cool, let me go do passages on the AMC portal, my anxiety had dropped. For you with an exam coming up, I'd encourage you to really be conscious about taking breaks throughout your day and blocking out your time and making sure your breaks are disconnect breaks, where when I studied, for example, I chose a place that had open lighting, as you can see, because I knew that would keep my mood up, keep me energized. And then I would take breaks, right? I'd study from like 9 to noon or 9 to 10 or 9 to 11, whatever time it was. But then I'd take legitimate 15-minute breaks where I'd go walk and clear my head, come back and do it again. Because the challenge is that you're going to do the same thing for 10, 12 hours a day for 2, 3, 4, 5 months a year at times. And, being able to learn how to reset is going to be a good thing for you on test day, because you have to reset 3 times on the test, right? Yeah. Between all 3 subjects or sections. Does that make sense? I know it's not that great of a piece of advice, but meditation and do you do any mindfulness breathing exercises, meditating exercises?
Not really. I've been told to, and I have the apps. I've just like not gone into it.
I highly recommend. I was an engineer. I have an analytical mind. I was like, no way, it's just too fluffy. Like, what? You breathe. And then I tried it. And what I tell people, I do a mindful exercise. I've done a mindful exercise before every exam I've taken since 2017. And it's all about clearing your mind of the things that don't need to be there. And on the MCAT, this isn't really a me thing. This is like a Jack Weston thing. So I'm not going to like copyright, but the moment you get something that comes in your mind that is not MCAT, you've already messed up. Right? Think about how many times like you allow your mind to be like filled with like lunch, anxiety about the exam, which is not even today, right? Anxiety about the section you already did, which you can't do anything about, right? So I personally believe in mindfulness. So that's my own 2 cents, more opinion than anything.
Okay. Oh, great. No, thank you.
Anyone else? I think there's 2 people. If not, I really want to thank you all. It looks like.
Question for you again. Sorry.
Yeah, go ahead. Good.
So this similar checklist is going to be applicable to all sections of the MCAT?
No, not at all. Not at all. So there's actually 2 checklists in here, and if you notice, it's about the passage, right? The informational passage is the most common passage in the Chem Phys section. And so you notice this is a slightly different checklist. It's a little modified because the way the information is presented is going to be different. And then this, I mainly use this checklist in Biochem, but remember in Biochem, the experimental passage is the most common passage type. Right? So even in cars, you're going to have a different model of looking at things, right? So again, guys, like, the big thing I can tell you is there's no magic bullet, right? The same thing I said about like Kaplan, TPR, they're all decent books, right? If they weren't, people wouldn't keep buying them. They just have to be right for you, right? So it's the same thing with like the different checklists and strategies. They are only things that have been shown based on them working on like for some people. So even from a baseline, right? Remember, like, you gotta, 1, personalize it to yourself, and then 2, just gotta be very careful when you take strategies from other places, not to apply them in the wrong setting. Because I know I was guilty of taking like car strategies that I learned and applying them to the wrong passage types and them not working. So it looks like we've got about 18 more people still in here. Anyone else have any last minute questions? Because if not, I'm going to go ahead and end the meeting for us. And in terms of follow-up, I will just be sending out emails probably Monday, if not Tuesday at the latest.
Thank you so much.
All right, awesome. Well, it's been a pleasure. I absolutely love getting a chance to share the few things that I've learned and picked up about the MCAT, about medical admissions, and about med school life, the little that I know after 2 years, or a year and some change. So I will probably be doing another webinar, or my colleague actually is going to be doing a cars-based webinar later on this week. So check that out. And if I have your email, that's great, because I'll just ping you to kind of update you on the dope things that we're doing.
Is there a link to sign up for the cars webinar?
I'll just send you, if I have your email, I'm going to just be sending out a email, and whatever Facebook group you heard about the session, I'll post in there as well.
Okay, thank you. I just left my email here, so thank you.
All right, awesome. Thanks, guys. Have a good one, and good luck studying, everyone.